picture I showed, there's also the possibility that two alleles are incompletely dominant to each other, which means the heterozygote looks like a completely different phenotype than either of the other two phenotypes. But we're not, yeah, so that would be like a white flower and a red flower results in pink, right? It's not red, it's not white, it's a completely different phenotype. Whereas codominance, you actually see both traits. That would be like a striped parent and a spotted parent are crossed, you get striped and spotted. It's not like completely different. It's not zigzags, that would be incomplete. If both show up, it's codominant. If something completely different shows up, they call it incomplete dominant. It's just the way those alleles interact. Oh, she's down there. Yeah. No. Right, so the blending inheritance said that pink now is a new entity, and if we go pink on pink, it's always pink. And we know that's not true, right? Because if pink was the heterozygote, and we do hetero on hetero, we know we go back to having that 3-to-1 ratio of phenotypes. So that's what sort of threw blending to crap, right? Blending suggests that this is now an entity that can never be separated. Pink will be pink, 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 pink. But they're not. They're complete particulate units, and when that white pops back out, and a purple, actually it's 1 to 2 to 1, sorry, yeah. The purple, pink, this is purple, pink, and then white comes back again. Right? So blending, but that's where that hypothesis came from, because sometimes white sheep, black sheep looks gray, but then gray on gray will give you white, gray, and black again. And there was another question? Yeah. Right, that's just what people were seeing, and so they thought, oh, somehow, and especially with humans, because we have a billion different features and traits and so forth, and kids look like a blend of parents a lot of times, right? And then their kids look like a blend of them, which looks like a blend of the parent, and so that sort of misconception was passed, like somehow the genes were combining in, or whatever it was that was causing it was changing and combining each generation, and they're not. It's just how they interact with each other. So yeah, there's a lot of other crazy stuff going on in genetics, but we're just doing the basics in here. Okay, and then Rh factor is just the positive and negative. That's a completely different gene. It's back to only 2 alleles under complete dominance. So it acts just like any other gene. It's just another protein on the surface of your blood cells. Okay, so negative is recessive. So if you're negative, right, that means your phenotype is you're negative. So I'm A negative. I don't know what my genotype is for the A. I could be homozygous A or heterozygous A, but I know I'm homozygous recessive for the Rh factor because I'm Rh negative. Whereas if you're a heterozygote, you're positive, you're Rh positive, and obviously if you're homozygote, you're Rh positive. Okay? So if you're AB positive, you know your genotype for your blood type, but you don't necessarily know what your genotype is for the Rh factor. You could be homozygous or heterozygous. But if you're AB negative, you know it for both. Right? Your genotype for your blood type is AB. Your genotype for the Rh factor is negative. And so then you can throw the blood type into Maury-Povich also, right? If somebody's, right, if the baby is negative and mom's positive, and we don't know what dad is, right? Obviously mom has to be heterozygous in order to have a baby that's negative. And then if, right, dad can't really get away with it for either. If he's positive, he could be that, and he could be the father. And if he's negative, he could be that, and he's the father. So there's no way to get rid of them. Old talk show sleazeball host guy. It's like on old... I know, but he's on like at midnight or 2 a.m. on some... Oh, is it still during the day? At the gym he was on? All right, go to the gym, people. Watch some good quality TV. Yes.
The incomplete dominant and co-dominant, is it considered homozygous?
Heterozygous. Always hetero, yeah. Those two classes are always hetero. So if mom's negative and baby's positive, right, we know mom's genotype. We don't know, right, what this other allele is for this baby. And so again, right, if the dad's negative, this still could be negative. And if he's positive, that could be positive or negative. So only sort of with a combo of parents, right, this guy, the positive had to come. Oh shit! I'm fucking this up. Had to come from the dad, right? Because mom can't give a negative. Mom had to give this one. The positive had to come from dad. So if one of the guys backstage are negative, then they're off the hook because they have no positive to give away, right? Because two parents that are both negative can only have negative offspring. So there's no way that he could be the dad because the baby is positive. It's just a protein that sits on the surface of a blood cell. It's named Rh because it's one of the proteins that causes a huge immune response when you do transfusions to other animals. When they were originally doing xenographic blood and trying to do transplants from monkeys to people or chimps to people, like if you try to give rhesus monkeys, which are one of the laboratory animals, human blood, they have a huge immune response, and it's to that specific protein. So they called it the rhesus factor, so the Rh factor. It's just a highly immunogenic molecule, and so that is one of the things, like you can't give somebody who's negative positive transfusion because you will create such a huge immune response to it that you actually have anaphylaxis and die. So when they were doing those, like why sometimes when you transfuse from you to you, it's fine and they live, and other times they have this huge anaphylactic shock and die. And so that's how they found all the different blood types and the Rh factor. There's tons of other proteins on the surface of these cells, but none of them cause that huge immune response, right? So for just getting a blood transfusion, you want to match the type and the Rh factor. So if you go to the emergency room and you're bleeding out, they're going to give you O negative, because everybody can take O negative. Because O means no proteins on the surface for the type, and negative is no Rh on the surface. So anybody can get O negative. Okay? So that's the emergency room blood type. So if you're O negative, the Red Cross is constantly sending you information, please donate, please donate, please donate, because you're the universal donor for emergencies. If you go to the ER and they're like, oh, you need to have surgery, they're going to blood type you, and then they're going to, if they need to transfuse, they're going to give you A positive if you're A positive. They're not going to waste the O negative when they know what you are. But they will blood type you every single time before a procedure. Right? Let's say you go in and you have a biopsy. They blood type you before it. If you go the very next day and they do another surgery, they blood type you again. Because if they make a mistake, if the nurse wrote down the wrong type on your chart, you're dead. So it's not worth, right? So it's irritating. O negative can give to everybody. O negative can only receive O negative. Right? AB positive can receive from everybody because they've got everything. But they can't give to anybody but AB positive. right? So they always want to give you your exact blood type when they can. If it's an emergency, they don't have time to type you, you're going to die because you're bleeding out, then you get O negative. But otherwise, if every little thing you go in for, they type you because they don't want to just trust that chart because it's so important. Yeah? Anytime blood is donated, that's all screened for all of that stuff. So unless you get a transfusion in another country where they don't screen, like the blood is very safe here, right? All the retroviruses are tested for, HIV, HTLV, all of that, and all the hepatitises. Now, before hepatitis C was even known to exist, yeah, some people had it and they gave it to other people and transfused 20, 30 years ago, right? Same with HIV. When we didn't know it existed before 1980, how could you test for it? So, it's within the realm of possibility, some emerging viruses out there that if you get a blood transfusion, you're going to get that. But everything that we know about is screened for. So if you're having a planned surgery, you give your own blood, and they stock it for you in case of emergency. You're just getting your appendix out or just something, non-emergency. You give blood the week before. They store it just for you. You have your surgery. If you need it, they give you your own blood. Right? If you don't need it, then it donates into the pool. They tested it for everything, blah blah blah, and then it'll just be donated. So it's safest, to give your own, or you can have friends come and donate just for you. You're going to need a transfusion during the surgery. You can have people come and that's your blood type come and donate. They screen it all, and then that's what they say for you. Okay. Oh my God, we're behind. So we did a lot of uploading. So that's good. Okay, so I posted that little sex determination video, so you guys can watch that. And it's essentially just, right, we talked about it before, eggs are only X's, sperms are X and Y. So, Dad, it's your fault if